Hold on, before we start, Margaret, do you have a plug for your computer? It says the power's... Oh, that's not yours, that's there. Oh, it is yours. Hi, everybody. I just see a low battery message on our computer. Somebody gonna help me fix that? Okay. I don't know who. Okay, somebody's got it. Okay, good. Yeah, you don't want that in the middle of the lecture. Hi, everybody. I'm John Mulcahy. I'm the president of Carnegie Science. Thrilled to welcome you all here today. It was very exciting to see we have so many new people. This is your first time here. For those of you who don't know a lot about Carnegie, we work kind of in three general areas. We work in life science, biology, as you're going to hear about tonight. Yeah, it's still, let me see. We work in astronomy, and we work in earth and planetary science. And as Erica mentioned, we've had an astronomy series here at the Huntington for many years, 23 years or something like that, Eric? I can't remember now. And in fact, Carnegie has been in Pasadena since 1904, when George Ellery Hale came here from Chicago to establish the Carnegie Observatories, which is our astronomy division. But the exciting thing that's happening is we are in the process of building out our biology effort here in Pasadena, so it'll be both our astronomy and biology here in Pasadena. So that's super exciting. And by this time next year, yeah, we're still for that. Yes. And by this time next year, we hope to have that all of our biologists here, so that's going to be very, very exciting. Now, tonight's speaker is going to give you a sneak peek of what the sort of work that many of those biologists do. When Margaret McFall-Ngai is here to talk tonight. She came in 2022. It's kind of our first hire here in Pasadena. But Margaret has a long history here in Southern California. She actually went to, I think, middle and high school here at Immaculate Heart. Some of you will know. It's kind of near my house over in Los Feliz. Yeah, so she's a local. We're excited to have her back. And after that, she also did all of her degrees here. University of San Francisco was her undergraduate degree, and then her PhD at UCLA, and then completed postdocs at UCLA and UC San Diego. She also then had a position as a faculty member at University of Southern California, USC. So she's really hit almost all the schools here. And she also has had appointments at University of Wisconsin, and then eventually the University of Hawaii, which is where she was before she came here in 2022. Margaret has many, many, many honors here. She's a member of the National Academy of Sciences, which is really kind of the biggest award one can give to a scientist in terms of societies. She's also a member of the American Academy of Arts and Sciences and the American Academy of Microbiology. Among her many awards, she got a Guggenheim Fellow in 2009, and she was a Caltech Moore Scholar from 2011 to 2013. So really a very established and exciting career. What you're going to hear about today, I'm not going to give her talk. They wrote some stuff for me to read, but it's kind of giving her talk, so I'm going to avoid that. Let her tell you about these bobtail squids, which are super exciting. So with that, let's bring Margaret out. Welcome her to the stage. I think you have power now.
So can everybody hear me? Okay, it's giving me some feedback. So thank you very much. First of all, I'd like to thank Carnegie and John and the staff for the opportunity here. It's great to see Carnegie, the Carnegie champions and the Carnegie, people, members of the trustees that were able to attend today. I'd also like to thank my high school buds. My high school buds are virtually coming in, as well as some of them are here in the audience, and some very, very long-time friends are here, and I'm a Southern California girl, and so lots of people I know here. And I also want to say a big thank you to the students and postdocs from the lab, from our lab, who could make it tonight. It really means a lot. So as the title suggests, I study symbiosis, and that is I study animal bacterial interactions or animal microbe interactions. It's kind of a new frontier field. I'm really excited to tell you about it, but it's really important for you to understand why this is a frontier field, where it came from, and how we're developing that frontier. So I'm going to start for the basis of the revolution. Why is this now? Why is this happening now? And some examples of its impact in symbiosis. I'd like to start off, though, by saying that, that I am speaking for the Biosphere Sciences and Engineering portion of Carnegie, but in fact, there are the observatories and Earth and Planetary and Planets Laboratory, and it's a very, very, very exciting time to be a part of Carnegie, because the president of Carnegie, John Mulcahy, has recognized that this is the time to integrate these three groups together, and I'm very happy to be a part of that. What we're doing in Biosphere Sciences and Engineering are a series of things that I've listed here. History is such a great teacher, and so we are interested in the planetary history and what that tells us about the evolution of life forms. We're doing patterns and mechanisms from molecules to ecosystems. Biology used to be very, very siloed into, you were either a cell and molecular biologist or you were an ecologist or evolutionary biologist. And the new wave is to look at ecological patterns, ecological patterns, and describe and discover the molecular mechanisms for those patterns. And then finally, down here, life across diverse and changing environments, particularly with the challenges that we face today. So the topics here will be the current revolution, like I said, symbiosis between animals and microbes as a common theme in the biological world. It used to be, I started working on symbiosis as a graduate student, and it was the biggest backwater field going. I mean, there was nobody studying, very, very few biologists studying symbiosis. Now it's huge. And so I'm going to tell you about that. And what the study of symbiosis has told us, of one symbiosis, the one we work on, is telling us about the dynamics of symbiotic systems. So let's get started. So what I'm showing here is I'm showing that there's a big sea change in biology. It's absolutely huge. And what a better understanding of life's diversity is unifying biology in ways that we haven't been able to unify the field. And it's very, very exciting time. And this is due to a technological breakthrough that I'm going to tell you about. And the ultimate goal at this point in the history of biology is to reshape it with all of this new information that we're gaining. So, how come this isn't working? Oh, there we go. Whoop. Went too far. Okay, here. So the thing is that for over 2,000 years, for over 2,000 years, biologists categorized the biological world based on what you could see, either with the unaided eye or with the aided eye. So it starts off with Aristotle, with just the unaided eye, and then Anton van Leeuwenhoek developed a very simple microscope, and then way up in the 20th century, we had the invention of the electron microscope. And with the invention of the electron microscope, we could see organisms in a different way, a little bit more closely. So this is the way organisms were categorized during these times. So there were things you could see, animals and plants, that's what Aristotle saw. And then Anton van Leeuwenhoek's simple microscope allowed him, what he did was he took a cheek swab and he put it in his microscope, and he saw these little, what he called animalcules. And so there were three groups. And then with the electron microscope, you could see very, very complex cells. And you could see what was going on. And so this guy here, Robert Whittaker, developed something called the Five Kingdom model. And it was based on what you could see with the electron microscope. So there were animals, plants, and fungi in the biological world. There were protists, which were single-celled organisms with a nucleus. And then there were bacteria. And those were things that didn't have a nucleus. They were single cell. And this was called the Five Kingdom model. So I'm wondering, how many of you had biology in college or high school? Wow, almost all of you. Okay, how many of you had the Five Kingdom model? Yeah, it's funny because when I ask that question, it's very, very common for people to have had the Five Kingdom model as the way they learn biology. Okay, so I don't know why this isn't working. But we needed to get a better idea of the relationship of animals and to get a more accurate picture of how the biosphere is categorized. Carl Woese, back at the University of Illinois, he reasoned that biologists needed an entirely new way to look at things that was a more direct reflection of what we are, and it's our genes. Our genes are what make us what we are, all the organisms on Earth, or what their genes are. So his idea came in the late 1970s, but what it requires is it requires the sequencing of the DNA. And the sequence, it will tell you what kind of organism it is. Okay, so he used genetic sequences to determine relatedness. Okay, but the problem was, in the 1970s, it was extremely slow and extremely expensive. And so his work went on very, very slowly, but he had the right idea for sure. So here's that technological breakthrough I was telling you guys about. A technological breakthrough, it's called next generation sequencing. And it was a method that allowed the cost of sequencing and the speed at which you would sequence to go from $6,000 a million base pairs, $6,000 a megabase, to 2 cents. Is that insane? And so that happened right about between 2006 and 2008, and it became really highly used over these last years. And so it's just an amazing breakthrough, huge, huge breakthrough. So I want to give you a prime example of how much this has changed the biologist's ability to understand relatedness of organisms. So a prime example is the sequencing of the human genome. And that project took from, like I said, it was slow, 1990 to 2003, and it cost $2.7 billion. It was called the Human Genome Project, $2.7 billion. In 2025, you can get your genome sequenced for $500. Okay, and then there are some companies who think they can do it for $100. I don't know about that. Now, so stepping back a bit, what I love to make this analogy. So a revolution through a technological breakthrough is like a new lens. So Copernicus here, this is astronomy, obviously. Copernicus had an idea, and that was that we were not geocentric. In other words, the planets didn't go around the Earth. Instead, he reasoned that we were heliocentric. In other words, the sun was in the center and all the planets went around the sun. And this happened before he had the idea, but he didn't have the way to prove it. So about 60 years later, along comes, Galileo. And Galileo, with his telescope, gets the evidence that's necessary to say, indeed, we are heliocentric. So this invention changed our view of the universe, and it changed our view of our position in the universe. So it was absolutely huge. So what happened with Carl Woese, we went from optics to gene sequence, and over 30 years, from the 1970s to the early 2000s. And then next-gen sequencing came along as a huge technological breakthrough, and it changed our view of the biosphere and has changed our view of our position in the biosphere. And that's what I'm going to talk about next. What happened was defining the microbial world became possible. So you guys, most microbes you can't culture in the laboratory. You can't make large numbers of them so that you can find out, what they are and what they do. Most of them are not culturable, and they're tiny, tiny, tiny things with no morphology associated with them. I'm sorry, there are microbiologists in the audience. They're round, or they're oval, or they're spiral shaped. So there's not a lot of information in the way they look. But what an international community swung into action, and one of the things was something called Terra Oceans. And this is the map of where they went all over the world to collect samples, and they collected, tons of samples and identified the bacteria in the water where they were sampling. The Human-microbiome project went on for 10 years, sampling thousands and thousands of people's microbiota. And then now the Earth's microbiome project is still going on with these characters are in charge of that project, and it's run largely out of UC San Diego. So, the biggest change in our view of the biosphere since Darwin, because this is what happened. Remember that 5-kingdom model? You've got this group here is now, when you put all the information together and all the sequences together and you align them and you determine their relatedness, you see that the microbes are, the diversity of life is overwhelmingly microbial. There's no question about it. And so here we are, and these eukaryotes here are just one branch of this type of microorganism, okay? This is huge. I mean, this is a huge change in the way we see things. And so one of the things that I like to say is it used to be thought that there were 7,000 species of microbes. That's the same number of species as there are snakes, okay? Which is ridiculous. Now there are thought to be billions, if not trillions, of bacterial species. Okay, so this thing's not working. So the other thing is that microbes are super, super abundant. So John would tell you, I think, this is the number that I got from the web, that astronomers estimate that there are 10 to the 24th stars in the universe. That's a lot of stars. The biologists estimate that there are 10 to the 28th microbes on Earth. The number of species we don't know, but there are 10,000 times more microbes than there are stars. So that's a lot of microbes. They are tiny. So we now are learning that the activity of microbes drive the form and function of life. Everything depends upon them as a foundation. They're pivotal to shaping the health of all corners of the biosphere, from the soil to the water of the oceans, to the atmosphere, to the bodies of animals and plants. They're absolutely critical for health. And the impact of this phenomenal diversity of microbes, like I say here, and the ecological implications continue to reshape the field of biology. It's slow. It's slow, and the recognition is slow because it is such a huge change in the way we see things. It's just huge. So let's look at this a little bit more closely and how we got here and what was the backdrop. So it is estimated that the Earth is about 4.6 billion years old and has been part of the universe for about a third of its history. So it's 4.6 billion years, so it's back up here. And so what's interesting is that life came up pretty darn quickly. And so life on Earth has been around for they keep pushing it back. I'm not kidding you guys. It wasn't 5 years ago that it was thought to be 3.7. And then it got pushed, and then it got pushed. And today I found that I have a little squiggle there because now it's back to 4.1. And so life is for nearly 90% of Earth's history. Okay, some major milestones. How did we get here? How did the diversity that you see when you walk out in nature, how did that all get here? Well, for most of life's history, all the way up here, it's unicellular, and most of it's bacterial. In other words, most of it's simple cells. And so you get the origin of life here. And then one of the things that happened was photosynthesis happened. They started capturing light. And when they capture light, when organisms capture light, microbes will do this, when microbes capture light or trees capture light, they emit oxygen, okay? So there was a lot of oxygen coming out into the environment. At first, that oxygen was sucked up by the iron that was in the rocks. But as soon as that was all oxidized, what happened was there was a big oxygenation event as the result of this continuing huge photosynthesis. Okay, so you get all this oxygen. And then what happens is there's a symbiotic association that starts between a bacterium and another bacterium to make that eukaryotic cell. And so that was way up here. And so what you've got are mitochondria. They're the powerhouses of your cell, came from a symbiotic association. Plastids, which is where in eukaryotic cells where photosynthesis happens, that was also a symbiotic event. And then you come up here and there's another big oxygenation event, something called the Cambrian explosion, which I'm going to tell you a little bit more. Then the first land plants come about at about 470 million years ago. And you can see the difference in planet atmospheric oxygen. We're now at 21%. At one point we were up higher in the Mesozoic, but 21 is pretty high. Okay, why oxygen? Oxygen is a powerful driver of energy production. And the reason I'm showing this rocket taking off is they use liquid oxygen as a way to convert the energy. So it's pretty phenomenal. Oxygen is a very powerful driver of energy production. And so that's one of the things that's obviously driven the evolution of the biosphere. So symbiosis, as I've alluded to just a minute ago, symbiosis is a principal mechanism of evolution of the biosphere. So you see it here and here in these. The Cambrian explosion, we don't know about. We don't know what drove the Cambrian explosion, but we know now that a lot of the animals were a lot of the Cambrian explosion was due to the presence of symbiotic associations. So I show here the humans in the last 0.007% of Earth's history. So we're minor players in this historical realm. Okay, the Cambrian explosion was an amazing period in life's history. So here, what happened was between 540 and 520 million years ago, all animal body plans were invented in this short, relatively short period of time. In this 20 million years, all animal body plans arose in the oceans. Everything was invented in the oceans. Different formats of structure and function. There are 35 types of animal body plans, 35 types of ways of being, everything from sponges to fishes. And then this occurred in the background of rich in bacteria, hugely rich in bacteria. So today there are about in today's seawater, when you guys go out swimming in Santa Monica, the bacteria has 5 million bacteria per teaspoon of seawater, okay? It's just teeming with bacteria, and they're all harmless, obviously. But they're very, very powerful in shaping the evolution of the organisms during the Cambrian explosion. So symbiosis is now known to run very large sectors of the biosphere. So here I'm showing a few. There's someone at Carnegie, a couple people at Carnegie who work on corals. That's a symbiotic association. And corals, of course, are very abundant for 30 degrees south and 30 degrees north of the equator. And they're very important for the ecosystem. Here in Antarctica, this is a dry area of Antarctica, and the only thing that exists, only life form that exists in this area of Antarctica are lichens. and lichens are a symbiotic association. And so here is a montane forest. Montane forests, actually, the water-to-land transition in plants, in other words, the ability of plants to colonize land, required symbiotic associations with fungi in the soil called mycorrhizae. And those mycorrhizae allowed plants to mobilize phosphorus from the soil. Otherwise, if we didn't have symbioses, we would have no montane forests if we didn't have mycorrhizae. And of course, I'm suspecting that almost all of you now know that humans have very complex microbiomes associated with them. How complex is the human microbiome? Well, symbiosis in many organisms is very complex. And so the human body, the mature human, has about 10 to the 13th or 10 trillion human cells. You're made up of about that many. You have somewhere between 10 to the 13th to 10 to the 14th cells in the microbiome, but many, 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 many more species than just the human. You know, you've just got one human, and you've got 10 to thousands of species of bacteria. And they're all over you, and here's a tree of them. And so the healthy human actually occurs as a nested ecosystem, as do all mammals and all vertebrates, actually. Okay, so think about the fact that there are all these cells. The cells number isn't that different, but the gene number is really different. And so because there are so many different kinds of bacteria, or microbes, there are 2 million genes in the average person that are microbial, and you only have 23,000 human genes. So no matter what you think of the person next to you, they're only about 1% human. Okay, so what do you do as a biologist with that kind of diversity and learn how it works? It's just too darn complex. So what biologists do when they are confronted with really complex things is they turn to model systems. And so I'm giving you some examples of really complex things. You go from a single cell here to 10 to the 13th cells. I mean, that's a miracle. In the brain, there are 10 to the 14th synapses. And in the human body, in symbiosis, you have 10 to the 13th bacteria. Very, very complex things. So what happens is that it's really common for biologists to, how many of you heard of the fruit fly? Yeah, the fruit fly. The fruit fly is teaching us about this. You know, it's just really simple, and lots of things are highly conserved across evolutionary time. And because microbes have been on Earth since the beginning of time, there's probably very, very few things as highly conserved as how animals and plants interact with the microbial world. So, why is this valuable? Well, the use of model systems from 1901, to show you how valuable they've been, 1901 to the present, there have been 16 Nobel Prizes in developmental biology, and 15 have been awarded to model systems, and 1 awarded to human research. So that's a good reason to support model systems, because they have, so what we've done with the animal I'm going to talk about is we ask a question. It's really simple, but the community doesn't know how to approach it in really complex systems. So what we do is we find an answer, and we say, look there. And that's what all the model systems do. In symbiosis research, as I said, it's very, very evolutionarily conserved, and so there are a lot of models that go all the way down to little hydra, which is like an anemone. And these are the experimental models that are really powerful model systems for the field of symbiosis. So, like this guy, the honeybee, you guys, every honeybee on every worker honeybee has 5 phylotypes of bacteria in its intestine. Invariantly, 5 species of bacteria, all around the world. I mean, that's pretty amazing. And so we're learning, you know, why we're losing our bees, and part of it has to do with the microbiome. But the stars of what I want to do is I just want to spend the last third or so talking about what we work on. This is Euprymna scolopes, the Hawaiian bobtail squid, and this is not a giant's hand. This is one of my technician's hands to give you an idea of the size of the adult animal. So this is endemic to the Hawaiian archipelago, and this is Vibrio fischeri, and it's the bacterial symbiote that lives with this squid. So I want to give some details about this, where this bacterium lives. The bottom line is that these bacteria live in the squid on the outside of the cells, just like your bacteria in your gut live outside of your cells. And so they live in very, very similar ways as they do in your intestine. And that's why they're so compelling for us to study. But what if you were to take this little guy, this casual guy, anesthetize him and dissect him open, you would find that there are two bilobed light two organs in the center of the mantle cavity right here. And if you look at one side of that, you would see that there are 3 crypt spaces. And the bacteria live in these spaces, these 3 crypts that are in the center here. And these bacteria make light for the animal. The light is used by this animal in its behavior. And so to make light, the light organ, or to handle light, to modulate light, the light organ has all of the structures of the eye. It has a lens, it has a tapetum, you know, like on the back of a cat's eye, the eye shine. And it has an ink sac, and the ink sac, so the organ is embedded in the ink sac of the squid. And the ink sac acts as an iris by the diverticula of the ink sac coming down, opening and closing and opening and closing. And it acts as a choroid to keep the light from going out the wrong way. So you can see here in these, this is a histological section. You can see the bacteria along here, and this TEM, I false colored these guys. This is the animal tissue, and these are the bacteria. So they're in their cheek by jowl, making light for this animal. So I want to show you how they look in there, what they look like when we're looking. Now this is something called a confocal micrograph. So we have labeled the bacteria with a green fluorescent protein, and the host material is stained. But here are all the bacteria, cheek by jowl in there. And look at that, how thin, they're in these thin layers that allow them to be within no farther than 1 or 2 cells from a host cell. So they're in very intimate contact with the host. Why do we like studying this? Okay, how many of you guys have seen My Dinner with Andre? I like asking this question. Fewer and fewer people every time. This was a great movie. Not a lot of action. They're just 2 guys at dinner. And it's really interesting because they start dinner and you get to know their motivations, what their relationship is with one another, and who they are, and what is important to them. The movie starts off with you thinking this guy's crazy and this guy's really sane, and you're in the movie thinking that this guy's sane and this guy's crazy. But the point is that if you have one host and one microbe, and you can drop in on the molecular conversation between those 2 partners, you can really begin to understand what a microbe would say to an animal cell. This is the human gut. This is madness. You know, try to figure out what the motivation is of any one person in that crowd. Okay, now what the squid does with this is this little guy is a night active predator. So what he does is he comes out at night, he emerges, he actually buries in the sand during the day, and he comes out at night to forage in the water column in the shallow sand flats of the Hawaiian archipelago. And so what he does with the light produced by the bacteria is he does something called counterlumination. He's matching a random sample of the light that's coming down from the moon and the stars so that he doesn't cast a shadow against the visual field of a predator looking up from below. And so, this seems pretty phenomenal, but I would have to say that there are large portions of the ocean where all of the animals counter-illuminate, because the light fields in certain portions of the ocean are so constant and predictable that it's driven counter-illumination as a major anti-predatory mechanism. And like I said, they bury in the sand during the day and are quiescent, and then they come out at night to forage. So that's what the light does for them. So this is Ned, my partner Ned, and me out in Kaneohe Bay, Windward Oahu, collecting squid. And so to give you an idea of what we do, we go 3 to 4 times a year to Hawaii, and we collect squid, only a dozen or so females and males. And then they will lay eggs for us in the laboratory. Now, we're interested in a whole series of questions with these guys, and they have something called horizontal transmission. So in symbiosis can either be vertically transmitted, that is the animal puts the symbiont in or on the egg and they participate in the embryogenesis, or they're horizontally transmitted. So humans have horizontal transmission. The embryos and the fetus is sterile and then starts to get the symbiont as they go through the birth canal and breastfeed and interact with other humans. And so that's called horizontal transmission of symbiosis. And so here you can see that there are stages. The embryogenesis, there are no symbionts. Then there's establishment, and I'm going to be talking a little bit about that, and development. So these guys undergo this trajectory, and then they have to be stable. They can't be rejected by the immune system, or they can't overgrow the host. So there's a stability created. Okay, so that's what we do. We study these phases of life. And here, the squid Vibrio symbiosis is transmitted horizontally, like I said. And what I'm showing here is I should take that off. I'm showing the adult female. She just came out of the sand, obviously. I'm the adult female, and she's laying a set of eggs, and we give them little hutches that the females can lay, and there are a couple hundred eggs there. And after about 20 days, they hatch out. So many of you, some of you have seen them. And after my talk, we will have a microscope with some baby squid for people to take a look. But here they are. On a given day, you can get hundreds of babies hatching. So there they are. Aren't they cute? Anyway, I'm just prejudiced. But these guys, they hatch out sterile, okay? And they have a tremendous challenge. And what is that challenge? So they have to get colonized by the light of their life, right? And so Vibrio fischeri is out there in the plankton because every day at dawn, the adults let go of about 90% of their bacterial symbionts into the water, and that creates the inoculum for the babies, okay? And so there are about 500 Vibrio fischeri cells per teaspoon in a background of 5 million in that teaspoon of other species. So this is a challenge to the baby to get the right one. In the absence of Vibrio fischeri, absolutely nothing gets inside of that light organ and grows, okay? So what you're seeing here is by 12 hours, they've selected the symbiont has gone through the tissues and is colonizing those crypts where they will live for their entire life. So there are some green-colored ones and some red-colored ones, one on each side of the light organ. But they're in pure culture at a tremendously high density. So they go from low density to high density. But it's a huge challenge. And one of the major questions we ask is, how do they do that? It's really fun. One of the ways that they do that, or one of the things that they have, is that the adult light organ looks like this, and the baby light organ looks like this. The adult light organ does not have these big fields here, and what these fields have is they have cilia on all over them that drive the water with the bacteria above the 3 pores where the bacteria will colonize on each side, here and here. So the juvenile light organ has structures that facilitate colonization. Each pore, like I said, leads to a crypt, and so there are 3 crypts here. Now, during embryogenesis, the most amazing thing is that the crypts don't all develop at the same rate. One starts very early, about halfway through embryogenesis, then the next one a few days later starts, and then the third one only comes on a day or two before the animal hatches. So they vary in maturity. So this is going to be important. There's the least mature is this little crypt 3. The most mature is crypt 1. So the crypts develop successionally during embryogenesis, and this turns out to be important. So I had a great postdoc, Tara Essick-Burns, who's now at Arcadia, and she worked in the lab, and she looked at this phenomenon of colonization of the crypts. And when the animal turns out she colonized the 3 crypts, and then she wanted to see how they behaved if you disturbed the population of bacteria, okay? And the way she did that was with treating them with antibiotics. Well, lo and behold, when she treated them with antibiotics, crypts 1 and 2, in other words, the most mature crypts, were completely killed. All the bacteria were lost. Crypt 3, they didn't even notice. Okay, crypt 3 the bacteria were fine in crypt 3. So that was a really, really interesting finding. And what she found when she looked farther at this was that after a little while, crypt 3 would mature by 48 hours, and then crypt 3 would act as a reservoir and would repopulate the other 2 crypts that were no longer populated. What a great idea. What a great idea to have one crypt with another job than the other 2 crypts at this point. Now, why is this going to be important? Well, I'm showing here a story about the likely impact of rising seawater temperatures on Hawaiian, on the symbiosis. So this is the water of Hawaii on September 14th, 2025. And you can see how hot the water is, okay? And so you can see this is Oahu, and this is where we work, and you can see that there's very warm water there. Vibrio fischeri, I should say, in culture never grows above 28, which is about 78, right, Ned? Yeah, in culture it never grows at that temperature. So, you know, we've got to start worrying with that. So I had a really wonderful student in my lab who worked in my lab for a few years, Evo Jock. She just defended this month. I was the co-advisor of Eve with Rui Rosa at the University of Lisbon, and she defended in Lisbon on the 9th of September. But one of her projects was to ask how different temperatures or increases in temperature might influence the embryogenesis of the squid. And so you see this is 25 degrees, 27 degrees, and 30 degrees. And she showed that the proportion of bobtail squid that hatch is lower at 30 degrees, and the developmental time is quicker at 30 degrees. That makes sense, because their metabolic rate is higher. The colonization success was down at 30 degrees. They don't get colonized as easily as they do at 25 and 27, and their survivorship goes down significantly. And so there's definitely a tremendous impact of temperature increase on these guys. And most importantly, the frequency with which they form a crypt3. The guys who were grown at 30 degrees, many of them, less than 50% of them, developed a crypt3. So, resiliency is compromised. And these squid lose their insurance card at higher temperatures. And so this is the kind of thing that can happen with global warming. So the possible result is that the bobtail squid and its symbiotic partner may be lost from Hawaii, but they may be able the squid is more resilient than the bacteria are at this temperature. And so they may be able to adapt over time. We're keeping our fingers crossed. So the take-home messages from my talk today are new findings show that microbes are critical to the health of all corners of the biosphere. Biologists have a huge frontier right now that can be, we're now enabled to explore that, and it must be explored. Symbiosis is a central theme in biology, and bobtail squid are cute. And I want to back out now and just say that John Mulcahy's vision is actually really important to the biosphere, because to identify the effective solutions to our immense environmental challenges that impact the biological world, we must bring to bear a deeper understanding of all aspects of our Earth. We have to integrate that information, so it's really important. With that, I'm going to thank my students, and so we have this Squid Vibrio. Ned and I started this about 35 years ago, and now we have a lot of academic children, grandchildren, great-grandchildren, and every year we have a meeting of the whole gang, and it's really fun, just so we don't compete. But I'd like to thank them all for all the great work, and of course thank Ned, who's been my partner all this time, and the funding agencies who have been so very generous with us over the years. So with that, could we have the lights out? So here's the guy burying in the sand during the day. You cute? Okay.
Thank you, Margaret, for an amazing talk. We are going to take questions. I'm sure we have many. We have microphones on the back, so we ask if people can go to the microphones so people online can hear. So I'll give people a minute to do that. Yeah. We have our first. Go ahead.
I think you mentioned that most recent evidence seems to indicate that life may have emerged around 4.1 billion years ago. Was this life? Did it use like contemporary A's, T's, G's, and C's, or did it use different types of bases?
Yeah, that's a very good question. There are a lot of questions of that sort, and they're very hard to resolve because it's oftentimes the evidence is very fragmentary, and so as far as we know, those bases were the foundation of all life and continued to be, and so there's no reason to think that wasn't the case. There are all kinds of things going on now, though, that DNA has a certain way that it goes, right? In a certain direction it goes, and it goes that way in everything that has ever been studied. And I have a colleague who is asking the question of what if we look for nucleic acids that are going in the opposite direction? And we've never done that. And we don't know. There may be a whole set of life out there that we don't know about. But thank you for that question. It's very, very interesting.
Wonderful talk, very inspiring. So the bacteria, the antibiotic resistance in the CRISPR-3 reminds me of something about biofilm. So inside biofilm, there's certain structure in the biofilm that can create this resistance to antibiotic. So I wonder for these different CRISPRs, for those, do they have different biofilm structures?
Yes.
Okay.
Yeah, that's a very good question. And in fact, you're right on. So CRISPR-3, in CRISPR-1 and 2, the bacteria are right up against the animal cells. They're in very close and intimate association. In CRISPR-3, they're oftentimes they're held at a distance, which looks like a pretty good biofilm.
I see.
Yeah.
Thank you.
One more we get.
What are the leading candidates for the messaging system that drives the diffusion of the microbes to the symbiotic relationship?
Okay, so that's a great question. So what happens is when the animal hatches, it spews out a huge amount, from that ciliated surface, it spews out a huge amount of mucus. That mucus contains antimicrobials that eliminate all gram-positives. Okay? Then what happens is it goes down and it begins to gather above those pores, and Vibrio fischeri adheres to the cilia. Nobody else adheres. Just Vibrio fischeri adheres to the cilia. It tells the host to pour out more antimicrobials. More antimicrobials get poured out. And so what we think, and eventually Vibrio fischeri is the only thing that is being gathered outside after a little while. Then one of the other things, so the host has changed gene expression to send out these antimicrobials. The other thing it does is it sends out a chitinase. And that chitinase breaks down the polymeric chitin that's in the mucus to the chitin dimer. And the bacteria, they turn on the genes associating with recognizing that chitin dimer, and they chemotag, the light organ puts a gradient of chitobios out, and the bacteria follow the gradient into the organ.
Amazing.
It's you know what,
Thank you.
we couldn't imagine anything more complex. It's one host and one microbe, and it's super complex.
The animals and plants only constitute a small part of the biosystem.
Yeah.
What does it take to wipe out all the plants and animals?
What does it take to wipe out all the plants and animals? Well, it depends on how hot it gets. So we're in the 6th major extinction, I believe. We're in the 6th major extinction at this point, and it's happening more quickly than the other major extinctions happened. And to wipe all of them out, I don't know. On those major extinctions, they wiped out up to 90%. I think it's 90% is probably the KT boundary. Any geologists in the group? Yeah, something like that. Yeah, so to wipe out all the animals and plants, I have to say that the microbes are a lot less susceptible. And one of the reasons is because their generation time is so fast, super fast. Like some of them is less than 20 minutes or 20 minutes. And so they can adjust. There can be selection that goes on and on and on in microbes. And that's one of the reasons why they're so resilient. Whereas our generation time is so long that, yeah. I don't know if that helped or not, but.
Thank you, it's a great talk. Here comes my question. So symbiosis always defined as the mutual beneficial relationship. Oh, no.
Yeah. You want me to tell you why? So I adhere, and most of the sort of traditional symbiosis people adhere to the definition of symbiosis as two different organisms living in close association. And there are 3 types that are defined by the fitness of the two partners. One of them is mutualism, like you were saying. Mutualism is when both benefit. In other words, their fitness is, they both benefit. One is neutral. One occurs where there's one benefits and the other doesn't, it doesn't care. And then the last one is pathogenesis, where one benefits and the other loses. Yeah, and so they're all types of symbiosis. But you're absolutely right that the word symbiosis had traditionally, by the people who study beneficial symbioses, had been called symbiosis, and they defined it in that way. But the pathogenic microbiologists think of symbiosis as being mutualistic.
Okay, so in this case, they will be more considered as the parasitism, or what kind of benefits for the viable to set up a symbiotic relationship?
I have teased Dad and told him that they're slaves. But, they get a lot of food from the animal, and their populations grow a lot. And so that's the benefit that they would get. Now, one of the reasons why symbiosis is a term that is really powerful is that it doesn't have a fitness associated with it. It's just symbiosis living together. And one of the reasons why that's valuable is it's very hard to determine whether or not, you see what I mean?
Yeah, I agree,
whether or not those partners are benefiting or not benefiting. And you hit the nail on the head there when you said, how do you know they're not slaves? And so, it's a really, really interesting sort of thing. Now, I'm part of something called the Arturo Casadevall school, and he's a very famous microbiologist. And he says that there's no such thing as a pathogen, there's no such thing as a mutualist, there's no such thing as a commensal, as they're called, and rather it's a state. Commensal, you can have a mutualistic state going on, you can have a pathogenic state going on. The bug itself is not a pathogen, and the reason is because Candida albicans is an example of a microorganism that everybody in this room carries.
Okay.
And when you're not sick, it's probably very important for you to have it.
Yeah.
But when you get sick, it's not a good thing.
Yeah, thanks for answering that.
Hi, I was just wondering, how do the squids, do they just have really strong eyelids, or how do their eyes not get irritated underneath the sand?
That's a good question, and I've never thought of it. Because there are a lot of organisms that live, a lot of invertebrates particularly live in the sand, and I can tell you that this guy, the males of this guy have one symbiont, and that's Vibrio fischeri. If you look on their skin, so if you look on our skin, we're full of bacteria that have co-evolved with us. These guys, you can look at them under a microscope with stain that will tell you whether there are bacteria there, and if there are, whether they're live or dead. They're always dead. You don't see any bacteria there, and if you see the occasional one, it's dead, because they have huge amounts of antimicrobials in their skin. And so what I imagine is that that the sand itself probably doesn't. They had to have selection so that the sand itself didn't abrade their eyes. Their eyes had to select for that. And I can tell you that if you came to our lab and you looked at where the squid live, they live in little condominiums about that big, and there's about that much sand in the bottom. During the day, if you look in, you can't see them at all. They bury completely. Yeah, so they must have, over time, figured out a way to keep their eyes from getting messed up.
Thank you.
Sure.
Thank you so much for your talk. It's fascinating to think that these squids are so selective with their microbe partners. And I just have to ask, is this system present anywhere in human biology? And to what degree do we choose the millions of microbes that are in our bodies?
So you absolutely choose. And so during embryogenesis, the animal lays down chemistry that is very specific to the microorganisms that are human associated. And so you don't take in, when you eat a banana or something like that, you're taking in all kinds of microbes. But the thing is, you have a microbiome, and it will change depending upon what you eat, but it's pretty stable otherwise. And the fact of the matter is that, yeah, I mean, there are, and it goes through, in humans, it goes through a tremendous succession. So in babies, there's a certain set of things that are chosen, and that matures, and the microbiome matures somewhere between 2 and 4. At the same time, the immune system matures. And what's really interesting is that one of the reasons why children can't recover from starvation, babies can't recover before the age of 2 or 4. It's very difficult to get them to recover from starvation. They just spiral down. Is because their immune system and their microbiome are not mature. And that was shown by Jeffrey Gordon. And so, in older people, you can go on a starvation diet in a prison or something, and then you can recover. And so, the microbiome is really, its establishment and development and there are a couple of things that are really important is vaginal delivery. You should have normal vaginal delivery because the microbiome is normal. The baby gets it from the birth canal. It starts the right stuff. And if you don't have that, the microbiome isn't normal even at the first age 1. 12 months old, the baby's microbiome is not normal. And it's also true of breastfeeding. Breastfeeding is extremely important.
So if I understand correctly, the microbiome itself is choosing what it allows in after we get that first template. Am I understanding that right?
No, I think it's a reciprocal conversation. It's definitely reciprocal. So Jeffrey Gordon showed that during embryogenesis, there's a certain enzyme that is turned up, and it makes a sugar that the bacteria like when they're coming down your gut. If you don't have the bacteria present, the animal turns off the enzyme.
Thank you so much.
Sure.
Let's do one last question over here.
Hello. Early in your presentation, you were talking about like model systems, such as like fruit flies or even like computational models used by researchers to provide like insights into human health. So like, other than squids, do you have a personal favorite model system finding that you think was interesting?
So yeah, so that little anemone, that little hydra that I showed, so that is studied by a guy at the University of Kiel in Germany named Thomas Bosch. And Thomas Bosch showed that that hydra can get cancer. And one of the things that's known is that most tumors have a microbiome associated with them. So hydra gets cancer, and it gets these tumors on it. If you look at that tumor, it's got bacteria in it. And so what was interesting about it was he decided to take and make germ-free hydra that didn't have any. They typically have lots of microbes on their surface, and 18 species. And so he made them germ-free so that they didn't have any. And he gave them the bacterium that caused cancer, and it didn't cause cancer. And he was like, wait. So what he did was he took the different, one at a time, those 18 of the microbiome, and he found one that if that was present, it enabled that bacteria, that other bacterium that causes cancer to cause cancer. Yeah. So there's a lot of fun. That was one of my faves.
Thank you.
Excellent. I know we can thank Margaret now, I guess. First. For a wonderful talk. I know there's more questions. If you want to come up, I'm sure she'll be happy to answer them. If you are not on the Carnegie Science distribution list, go to carnegiescience.edu and sign up. It'll let you know about lectures like this. We have an open house at our astronomy division observatories on October 12th, things like that. Lots of fun. So we encourage you to do that. And with that, don't forget to go see the baby squid.
Yeah.
They're out there somewhere.
Baby squid.
So go and have a look. Thank you, everybody. Have a good night.